Welcome to another episode of Whose Gene Is It Anyway? I'm your host, as always, Justin. Welcome to the show. Today's going to be real fun. So, if you saw my post on Twitter or my post on the community page thing on YouTube, you'll know that I'm doing streams a little bit differently. So, previously, when we were doing these Whose Gene episodes, I've been drawing an idea out of a hat. And while this has been very fun, it's kept me on my toes, it has led into some issues. Primarily, there'll be like these huge chunks of time where I'm basically just researching things, and it's not particularly fun to watch. So I decided that we're going to do things a little bit different. Also, the other issue is because I don't know what the topic is, I can't make a custom thumbnail, and so the thumbnail was super generic, so people weren't watching as much as they otherwise would. So I figured we're going to do something a little different. So I posted a poll both on Twitter and both on the community page on YouTube. And rather than, picking random topics, I picked some topics I was interested in and excited about, and I figured I'd just let you guys pick which one we do. So the options were tearless onions, non-browning apples, vanilla milkshake yeast, and magnetoreception. And you guys chose magnetoreception, so that's what we're going to do. So, I'm going to take you... So I did a little bit of research. This is something I was a little familiar with, and then on review, when I was going through the literature, I was like, oh wait, no, I was wrong. I thought this was going to be a lot easier than it is, but it ain't, so here we are. Okay, so magnetoreception. Let's talk magnetoreception. Actually, before we do that, there's a few little announcements that I got to do, and then we'll get into it. Thing number one, you'll see the donation bar here. So, as I mentioned in previous streams, I'm working on building a editing computer because the one I currently use is trash. So, yeah, we're fundraising piece by piece until eventually I end up with a computer, and then once I've got all the pieces, I'll do a stream of putting it together. So I've already got the motherboard that I just bought myself. We just finished fundraising for the CPU, and so both of those have been ordered and are on their way. And now we're on to RAM. After that, we've got the GPU, cooling, hard drive, then screen and stuff. But I mean, we're working our way through it, which has been really, really great. So, be that as it may, that's really cool. So if anyone feels like donating, that's where your donations are going. It makes my life a lot easier, and it's greatly appreciated. So thank you very much if you do that. Just so you're aware, only donations that go through the donation link in the description will end up affecting this bar. I don't know why it's like that, but it is. But anyway, so then, super chats go to, food, which I hear is important. Anyways, okay, so that's that. Announcement number two is I have a newish video on my second channel, which if you didn't know I had a second channel, you should absolutely go check it out. It's called The Taste Emporium. In fact, I can just show you. This is the Taste Emporium. If you haven't already, please go subscribe, check out the videos. I think they're very pretty. I think you'll like them. Okay, so those are all of the announcements. Now we can actually get into magnetoreception. So I have all these papers pulled up, and we're going to kind of work our way through them. And so let's just talk about this. So thing number one, what is magnetoreception? Excuse me. The short version is it's birds' ability. Excuse me. It's birds' ability, or well, it's not just birds. It's animals' ability to detect magnetic fields, primarily the Earth's magnetic field, because they use this not for finding magnetic rocks, but for navigation. So the birds are able to detect the Earth's magnetic field, and from that are able to navigate, which is why they're able to cross such ridiculous distances and not get lost usually. So that's the thing. Also, fair warning, shit posting will get you banned. Let's just get that out of the way at the beginning, because, whoo boy, is the comment section already spicy. So, that as it may. Okay, so magnetoreception. So there have been several papers on this. I mean, there's been many papers on this, because no one is particularly sure how they actually do it. Some people thought that maybe there were little particles of magnetite in their beaks, and so that was acting as the compass. But thus far, nobody's been able to prove that. Oh, thank you, Connor. Greatly appreciate it. So yeah, there. As far as they can tell, there was not magnetite in the birds, and that wasn't doing the thing. So it's like, okay, well, so if it's not magnetite, and the way that they figured this out was they stuck magnets to the bird's head, and then they were still able to navigate just fine. Which that is indicative, because if there was magnetite inside the here, actually, I'm gonna go magnetotactic bacteria. So, for example, this is an organism that has magnetite in it. They're called magnetotactic bacteria. And you can see they literally have these little granules of magnetite in them, and it allows them to line up with magnetic fields. So, if birds had this, okay, you're getting banned. So yeah, if birds had this, if you stuck magnets to birds' head, it would be permanently polarizing the magnetite into like one direction, and so this is kind of a problem, because then it would screw with their ability to detect the Earth's magnetic field, and they wouldn't be able to navigate. Except when they did this, the birds were able to navigate just fine. So it's obviously not magnetite. So it's like, okay, if it's not magnetite, then what is it? Oh, thank you, DeWall. I'm actually going to be... So, DeWall says that some scientists theorize that ancient humans may have been able to sense magnetic fields. We'll talk about that. Because, okay, so moving on past magnetite, it's like, okay, well, so if it's not magnetite granules, maybe it's something else. So, for a while... people thought it was this weird protein called MagR. And so MagR is found primarily in the eyes of birds, and it was also associated with other proteins. So basically there were several proteins as well as MagR that were suspected that maybe those were the magnetic sensing thing. Fuck. Y'all, chill the fuck out. Holy crap. Anyway, moving on. So, basically there were several proteins that they suspected that maybe those were the magnetic sensing things, and they thought that they were in the eyes of the birds. So the ones that were primarily suspected were this one called Cry4 and Cry1A. So these are proteins that typically are there. Normally they sense, or they're part of the blue UV-ish sensing system. Because there were some studies that showed that, if the birds were in darkness, they weren't able to navigate by magnetic fields. But then there were other studies that showed that there's various birds that when they're in complete darkness, they can still navigate. Somebody just kicked in. Oh, thank you, Mr. Roberts. Greatly appreciated, as always. Anyway. So the way that this was suspected to be functioning was actually really interesting, and so I kind of want to just talk about it for a moment, and I'm going to see if I can pull up a picture of it, because it's kind of weird looking. Yeah, okay, here we go. So, MagR is the sort of core, it's this yellowy core part of this, and Cry is the outside. So the idea was that these two proteins would interact, and they would form this sort of tube, basically. And so what would happen is... Oh, thank you, Exor. Greatly appreciated. Okay, so the way this works is MagR would associate with iron sulfide granules. So you would basically end up with this like long line of iron sulfide granules surrounded by these cryproteins. So the thing is, iron sulfide on this scale, like there's literally like 12 atoms here. Like this is not, like this image makes it look like there's a lot more stuff here than there really is. There isn't. The like 12 or 14 atoms worth of iron sulfide is not enough to actually respond to a magnetic field. So this is kind of weird. So it's like, okay, well, we've got these iron sulfide granules, and they're lining up like a compass, and they're surrounded by these cryproteins. How are they able to detect magnetic fields or align to them? Because the way this protein was found was through this study, where they checked all sorts of proteins in various animals, and tried to find ones that are magnetic and like aligned with magnetic fields. And so they ended up finding this one, which when you stick a magnet on it, it lines up with magnetic fields. So going back to the cryproteins, like, okay, well, how does that work? And turns out the way that these are not magnetic, they're not ferromagnetic like we think of with like a chunk of iron where it like automatically lines up with a magnetic field. They're what's called ferrimagnetic. So basically, the way this works is this is theorized to work. No one actually knows if this works properly. But the way this is theorized to work is the cryproteins absorb blue photons, and they use that energy to basically pull the electrons in the iron sulfide granules off a little bit. So this sort of spaces them out and readjusts the distribution of the electric field, which makes them much more sensitive to magnetic fields. So the idea is when there's blue light, it stimulates this protein complex, and it makes this thing line up with magnetic fields. Great. Here's the problem. When they found this, everyone was like, "Oh my God, that's got to be it." The link, Crazy Uncle Bob, thank you for donating. Although I don't know why, but the link doesn't work. Because somebody just donated through the link in the description, so it should work. I don't know what's going on. Okay, I'm gonna just tend to the comment section for a moment because shit be fucked. And then we're gonna get back into this because holy crap. Okay. There's another one. Alright. Y'all can stop being toxic, that'd be great, and we can focus back on the science. That would be lovely. Okay, anyways. Basically, this is a really interesting protein complex. Like, I mean, even if this isn't the magnetic sensing domain, or it's maybe not the sole magnetic sensing domain, it is a fascinating protein complex nonetheless. Here's the problem. This paper. So this paper is called Reevaluation of Magnetic Properties of MagR, and this other one. So these are other proteins as well that have also theoretically been detected as magnetic. The problem is, what this study did was they're like, okay, well, these proteins are great, but do they actually like trigger a neuron when a magnet is applied to them? You're getting hit. Fuck off. Bye. Okay, good. Yes, lovely. So, anyways, the point being, they actually found that the neuron spikes that they were getting when people had been studying these previously were essentially random, and they just happened to line up with the like magnetic polarization. And so basically what they're claiming is the other studies suck and just hadn't been rigorous enough, and so they were detecting neuron spikes, but not because of like the magnetic stimulation, but because neurons just kind of do that sometimes, and they just weren't kind of paying enough attention. So in short, magnetic proteins in eyeballs, probably not the magnetic sensing domain. maybe they play a role, but they don't play the role that people thought, and they're maybe not the main core mechanism. Cool. So that's that. So it's probably not eyeball proteins. So then we get to this paper, which I just found yesterday, and is fascinating, and frankly, I think is much more accurate. Is it? Is the stream frozen again? No, okay, good. Anyway, so this paper is fascinating. So it's called a putative mechanism for magnetoreception by electromagnetic induction in the pigeon inner ear. Okay, what's this mean? So basically, rather than a specific magnetic protein, this actually uses sort of like a magnetic loop antenna. So in radio, there's this concept of a magnetic loop antenna, which is basically what it sounds like. It's a big round, usually copper loop. And rather than detecting changes in an electric field, like a typical antenna, it detects changes in a magnetic field. So this is particularly useful for certain types of radio, for direction finding with lightning strikes. We've talked about this a little bit. And so the idea is when a magnetic field passes through, in fact, I'm going to just pull I'm going to just pull up a picture so that hopefully this makes a little bit more sense. See if there's a good one that illustrates the point I'm trying to make here. Okay, so either way, this is a magnetic loop antenna. The idea is, wow, that just got really bright. Give me a second. My light just went very bright. What the fuck? Oh, for fuck's sake. Just a moment, y'all. This is what I like to call why we can't have nice things. Okay, we're back. I moved my my lighting box before the stream, and it seems to have loosened my softbox. Okay then, moving on. So, anyways, this is a magnetic loop antenna. Let's see if I can see one. Yeah, okay, this is closer to what I'm looking for. Okay, so the what the fuck? No. Okay, fuck it. We're just gonna go with it. So the idea is with a magnetic loop antenna, if a magnetic field passes through the inside of the loop, it induces an electric current in the loop itself. Guys, fucking. So yeah, it induces an electric field in the loop that like circles around the loop depending on which direction the. Oh, thank you, Simlink. Greatly appreciate it. What the fuck? Okay. So yeah, basically, if the magnetic field goes one way, the electric current that is induced circles one direction. If the magnetic field goes the other way, the field circles the other direction. Matthias has a question and donated, so thank you very much. What is your question? Is it possible to create antibodies in bacteria or yeast? Technically, yeah. Oh, wow. Jesus. Okay, so to answer Matthias's question really quickly, is it possible to generate antibodies in bacteria or yeast? Technically, yes, but it's hard. And it depends on the exact style of antibody. Like a full antibody is hard. It doesn't necessarily fold properly. Mason just donated, and thank you very much. Greatly, greatly, greatly appreciate it. Okay. Now, back to magnetic loop stuff. So, basically, yeah, magnetic field goes one way, electric field, or like electric current spins one way, magnetic field goes the other way, electric field spins the other way. Okay? Cool? You're comfortable with this concept? I'm assuming you are, because you can't talk. So, I mean, yeah, this is literally, like, this is exactly how a radio receiver works, except that obviously biological systems can't function like a radio receiver, right? Wrong. They totally can. Okay, so inside the inner ear, and I think, yeah, okay. So this is the inner ear system of mammals. Basically, it's this is how we detect how, like, balance. Basically, like, this is sort of like our biological accelerometer, and it's this series of rings filled with fluid. And at the base of the ring, there's this little cluster of cells that are extremely sensitive. So basically, if you're spinning, right? Like, if you spin around really quickly. Oh, thank you, Timmy. Greatly appreciate it. So yeah, if you spin around really quickly, the fluid in these canals will literally flow around the ring, and it'll stimulate the little neuron bundle at the bottom. And so this is how we know, like, if you like turn your head really quick, this is how you're actually detecting the motion of that. It's through the flow of these fluids. But in this paper, what they posit is that in some animals, the cells that are in this semicircular canal, in that little bundle right at the bottom, are actually sensitive, not just to the flow of the liquid in the thing, but also if an electric field is induced in the ring. And it's literally a... Oh, thank you, Sporko. Greatly appreciate it. Since it's literally a tube filled with conductive fluid, it basically acts like a magnetic loop. And so if you have a magnetic field that's applied to that loop, it will induce a current in the thing, which is enough to trigger the neurons, and so this is how the animals are able to detect the magnetic field, since as they're flying, the magnetic field will be changing a little bit, and so that's enough to continually stimulate those neurons as new currents are generated, even though... So it's basically like the brain learns that, okay, well, I'm not tipping, but I'm getting this input, so it must mean magnetic field, right? Like, it's the kind of thing that, like, if you grew up always experiencing this, like, your brain would learn. So now the big question is, okay, great, this is wonderful. Why doesn't it work in humans? This is what this paper also posits. So it turns out that there's a specific voltage-gated channel, which, so for people who are not familiar with neurons, the way that they fire is typically what are called voltage-gated channels, and they can respond to all sorts of different stimuli. In this case, the one that they're interested in is a calcium voltage-gated channel. So basically, when a certain level of calcium is present, it triggers the thing. So in this case, this could be, like, the flow of the solution may shift the level of calcium in the ring, which is what's actually triggering the thing. It can also be through pressure. So certain structures can literally be reserves of calcium, and when you bend the structure, it literally, like, squishes the cells, and they kind of, like, fart out a little puff of calcium, which will trigger the neurons, and then that stimulus can be transferred to the brain. So the idea is, okay, cool. So it's a calcium channel. So what they found was holy shit, Genbox, thank you very much. If I'm correct, I think we just hit our goal. It's going to take a second for... Yeah, there we go. Holy shit, we hit our goal. Thank you. Fuck yeah, dude. Amazing. Greatly appreciated. Y'all are wonderful. Okay, so... fuck yeah, this is awesome. It's a good day. It's a good day. Okay, anyways, so getting back to it. So what they found was a particular calcium channel, which is basically only present in this inner ear system and in the retina, is different in things that can detect magnetic fields than everything else. So I'm just going to address this question really quickly. James, thank you very much. He says if you had to make yeast with several genes, like 8, would you make 8 different yeast or one that could do that? Yes, you could totally make one yeast express 8 genes. It's tricky and it's hard, but yeast are so large that they're actually quite capable of doing this. It's physically difficult to engineer. Okay, anyways, back to the thing. So basically, they posit that they basically found variation that was consistent between species that could detect magnetic fields and species that can't. So, for example, like humans do not have this extra little stretch of amino acids within this protein. So I'm going to make this a little bigger so you can see this. So this is basically a cartoon view of what this calcium protein looks like. So it's what's called a transmembrane protein, which makes sense because its job is to let calcium in or out of the cell, and when it does this, it literally changes the voltage potential of the cell, which triggers the axon to fire. Like it triggers an action potential. So this is the protein, and in species like humans, there's this chunk missing. I mean, it could be. It depends on how you look at it. It could be missing. It could be only present. Point is, in things that detect electric or magnetic fields, there's this extra chunk here, whereas in things that can't, it's not present. So this is why humans are. So we actually can detect magnetic fields a little bit. It's just very weak and it's not ingrained. And so I would actually posit that the same mechanism is happening, just something about this change makes this a little more sensitive so that we're able to detect it. There's actually a really fascinating Veritasium video where he went and interviewed some researchers who are studying this, and sure enough, when you put a human in the dark in a big magnetic coil, and then measure their brain activity, you can actually detect that the brain is responding to the change in magnetic field. It's just so minimal that we're not aware of it. Whereas in these animals, presumably this makes the protein a little more sensitive so that it becomes an almost conscious response. We don't know if it's conscious because no human has been able to do this, but it's the sort of thing where the animal can respond to it rather than it just kind of being a thing. Oh, Norman, thank you very much. Rose and petrichor smelling armpits. Neat, but also gross. Okay. Okay, so the somebody else. Oh, somebody became a member. Cool. Cool. So, point being, in theory, if you could modify this protein in the ear canals of humans to have this extra little thing, it would make them more sensitive. Thank you, Andy. Greatly appreciated. I will do right by you and by ramen. So, the idea is, if you could modify the cells in the ear canal to have this extra addition, you could either, I don't know if restore is the right word, but you could add or allow for sensitivity to magnetic fields, which sounds pretty fucking cool to me. So, yeah, 64 gigs of ramen. Nice. Ramen processor. Okay, cool. So now we know that this is the thing. So now let's talk about how we're going to do this. So we need to modify the cells in the ear, which is not trivial. So there's two different ways to do this. If this is the thing that... No, this is not the right thing. Okay, so then there is this paper, which says recent developments of AAV-based gene therapies for inner ear disorders. So what's important about this paper, and I'm going to basically just summarize it really quickly, is they're using a special kind of virus called an AAV to deliver the genes to the to the various tissues in the ear. Oh, Car Jesus, thank you very much. Greatly appreciate it. Actually, somebody obviously aware says, can shark use their electrophores to detect magnetic fields? And so what's really interesting is in this paper, you'll notice this picture of a shark here, because their sensory epithelium apparently uses the same protein in their sensory organ. So they're able to detect magnetic fields through an electrophysiological mechanism rather than a inner ear thing. So yes, they are, and they're able to navigate because of that, which is really fascinating. But in theory, they use the same protein, which is cool. Getting back to it. So this paper talks about modified AAVs to deliver genes to the hair cells. So this is to try and repair people's hearing loss, which is fascinating in and of itself. But their method basically relies on, and I wonder, yeah, okay, here's a good picture. So this is what the AAV looks like. Their method relies on physically modifying the shell of the virus to be more specific to target exactly that tissue. Now, because of what we're trying to do, I don't know if that's, I mean, you could do that, it would just be very hard. For the sake of discussion today, rather than this, we're just going to pretend that if you were going to deliver this, you would literally just have to stick a fucking needle into the area, basically, and just, put a little spurt of regular virus, unmodified, to deliver the genes to just kind of that area. Like, you just do it by dosage control and physical manipulation of where you're delivering the genes, rather than something that would be delivered globally and have to migrate to the right area. So, you probably wouldn't want to be conscious for this because it hurt, but doable. So the idea is, I'm going to pull this up again. You can see, so here's a little clump of cells. You would have to basically get the injection to essentially right next to this. But because of how we're going to do this, it shouldn't matter if the virus got into other cells in the area, because it'll only affect this clump. And we'll talk about how that's going to work in a second. So, the idea being, just to summarize, so what we've got to do is we need to slightly tweak one protein in this very specific location in the body to be able to make this magnetoreception ability thing happen. Seth just donated. Thank you very much. Seth, we'll come back to that in a minute when we actually do some of the genetic modification stuff. I want to actually finish just going through this preamble. Okay, so let's just summarize quickly. So what we've got to do is we need to deliver some new genes to exactly this area, and we don't need to we're not going to just apply a whole new protein, although I mean we could. I think what would be better is if we just modify the protein that's there, which means just adding this little sequence here, so that we can manage this. So I've already found 2 plasmids, which we're going to be using for this, because this is going to have to be a double whammy, just because it's easier to design that way. So the last bit of info that I need to share is this. So this paper is called Efficient Gene Transduction of Neurons by Lentivirus with Enhanced Neuron-Specific Promoters. Okay, what's important about this paper is they have found a series of promoters, which, as a reminder, if this is your first time tuning in to a stream, A promoter is the bit of DNA that goes before the gene and basically tells the gene when to start running. So sometimes they just kind of run all the time, or in this case, they only run in specific tissues. So basically, what we're trying to do is essentially dial in the expression of this protein change that we're going to make to exactly the right cell type, because we don't want this running just randomly, or you're going to end up with, like, you know, magnetically sensitive cells on your face or on your butthole, and nobody needs that. So we're just trying to keep things as tight and narrow as physically possible. So the important part of this is the name of the promoter that they found is called SYN. It comes from they found it in association with a gene called synapsin-1. They ended up making an improved version called eSyn, but for our purposes, we're just going to worry about, version original of SYN. So I found an AAV plasmid. Remember that AAV is the virus thing that we're going to use, that has this SYN promoter on it. In this case, it's just got some GFP, but that's fine. We can cut that out. But the important part is it's got this SYN promoter on it. Very cool. It also has some other stuff, which is not super helpful. In fact, we're going to actually get rid of all this. But the important part is it's got this SYN promoter. It's in an AAV. So the idea is if you took this piece of DNA and you mixed it with the other two that you used to grow... In fact, I'll just show you this. AAV 3 plasmid system. Okay, so let's just quickly review how an AAV works, just for people who haven't seen my lactose video or are familiar with this. To make AAVs in a lab, you use 3 different plasmids. So plasmids are little circles of DNA, just as a reminder. So you've got the one that you actually want to get packaged into the virus, and then you've got the 2 other ones that code for the shell of the virus. So that's what's on here. And then the helper plasmid, which basically handles copying of the DNA. So as long as all 3 of these are present in the cell, not only will it produce virus particles and package this, everything's getting copied. So you get not just like the amount of virus proportional to the amount of DNA you put in, but you get just as much virus as these cells can make before they die. And then what ends up happening is the viruses that are produced will only contain this piece, which just contains the gene you're interested in, and none of the replication stuff. So they become essentially just delivery mechanisms. So if you were to inject this into a patient, it would be able to deliver just this gene of interest and none of this crap. So once the virus opens up, it releases the DNA, and then that's it. There's no more virus happens. Like it gets broken down, and then that's it. So these plasmids, and the way this, basically the way you set this up is, there's these, they're called ITRs. Basically, what is that? Oh my god. Oh, chill. See if that's anything important. No. Okay. So basically the way this works is, and I can actually show it to you here, there's these chunks called ITRs. I forget, telomeric repeats. I forget what the I stands for. But basically it's just this repeating pattern of DNA. And there's one that marks the start, and one that marks the stop. So basically anything that's within, like between these 2 pieces, is what gets packaged into the virus. The virus doesn't actually check. It's just like, oh, well, the ITRs are there, just package. So basically the way that you do this is you get a plasmid like this where you've got ITR, and then gene of interest on the inside. And when you grow this in that triple plasmid system, only this part gets packaged. Like it cuts this crap off. It cuts here and it cuts here, and this part gets packaged into the virus, and everything else is ignored. So I grabbed 2 of these. So one of these has this SYN promoter, and a bunch of crap which we're going to cut out. And then this other one has ITR, ITR, and a Cas9. It also has this promoter, MECP2, which apparently is also neuron specific. So basically what we've got is two plasmids that are both AAV based. This one we're not going to modify. This one we're going to just use as is. I say we're going to use it if we're actually going to do this. We're fucking not. But theoretically, what we would be doing is you would be growing two strains of virus, one that has this piece in it, and one that has the thing that's going to be in here in it. And then you would basically apply both of these. So, what is this one? This, as you might be able to see, it says Cas9. Cas9 is better known as CRISPR. We're finally doing some CRISPR stuff. So, what we're going to do is we're going to use this protein, and so what's interesting about this protein, Cas9, in fact, I'm going to pull up a picture of it, is this. So, what Cas9 does is using what's called a guide RNA, it scans DNA for a piece of DNA that matches this guide. So, the way this works, we're going to have to basically design a guide, and at a specific point in the guide, and it's always the same point, like you, there's I'll show you in a minute, there's a system that lets you design these things, but basically, this is the bit that ends up matching the DNA, and then once you hit a certain length, it cuts. So the idea is we're going to make two cuts at the start and stop of the sequence. So if we go back to here, we're going to have to find this bit of DNA, make a cut here, and then provide an extra little bit of DNA that codes for this sequence. And when the mechanisms inside the cell go to repair the damage, because that's basically what this is, we're damaging a specific piece of DNA by cutting it open. When we do this, the cell is going to go, okay, well, the DNA is broken, this needs to be fixed. But there's a repair mechanism that allows you to provide an extra piece of DNA which it'll shove in the hole, basically. So what we have to do is essentially make, in this case, because we're just trying to shove something in a hole, like we can make one cut, which is fantastic, and then provide a homologous bit of DNA which will go into the hole and get repaired. And so it's not going to be perfect. This only will happen, say, 90% of the time, but the idea being is you should end up with more of these proteins having this new code, essentially, which should make it so that as the protein is, because the protein is constantly being generated and replaced as the cell grows and dies and this kind of thing. So as the next round of proteins are made, as these ones get worn out and broken down, they should contain this extra little fragment, and we should end up with the thing that we want, which is good. Okay. So now we know the game plan. So I'm gonna, after the stream is done, I'm gonna go in and post links to all of this stuff. And it'll also be on the GitHub, which I have not talked about the GitHub yet today. GitHub. Okay, this is the Whose Gene Is It Anyway GitHub, which I highly recommend checking out. All of the plasmids that we end up making end up in here. So if you want to follow along, if you want to have some of these made, if you want to play with them, they're available for you to do that. There'll be a new folder with all of the stuff that we talk about today, links to papers, this kind of thing. It'll all be here. As a reminder, I only ever post links to the actual paper itself. I don't post Sci-Hub links because I don't feel like fighting Elsevier, because fuck them. But just a reminder that if you want to get a paper and don't have access to it, Sci-Hub is the way to go. So just really quickly, somebody just donated, Cordy, thank you very much. And they asked, can we just use simple inorganic devices such as the magnetic loop antenna and feed the output directly to the brain to teach it to sense magnetic fields? I mean, technically, probably, but it'd be hard. And also then you've got to worry about like a physical thing breaking down, whereas with this system, it's biological. It'll constantly repair itself if anything happens. Also, so Cas9 is just the one we're using because it's available. It is not the best Cas protein. There are literally dozens and dozens of different Cas proteins, and some work better than others. Some will cut differently. Some make blunt cuts. Some make sticky cuts. Some just do little mutations. We're just using this because it's available. It is not necessarily the ideal thing. If I was going to use this on a person, which I'm not, but if I was going to, I would not use Cas9. I'd take the time to develop this better. We're just doing this for the sake of the discussion today. So let's start downloading some DNA and getting this all set up. So I'm using a site called Benchlink, which I can spell reception. Yeah, good enough. Okay, so I'm using a site called Benchlink, which is basically like a visual coding program for DNA. I actually quite like it. It works quite well. So before we do any of that, let's download this. And I'm going to download this as well, because, you know, why not? Gmail. Okay, so there's different file formats for DNA, which I realize that sounds insane, but here we are living in 2020. I prefer the GeneBank version. SnapGene also works. They basically all comes out the same. So I'm going to close all of this stuff once we have this open. Okay, so we want to import DNA sequences, choose the file. We want these two. Open, please. What are they going to... Okay, we're going to use this. Close. Okay, these are now loaded in. Okay, what do we got here? eGFP. Okay, this is not the one we want. We want this one, I think. No, actually, this is the one we want. We wanted this one. Okay. Close others. Okay. I had a bunch of stuff open. Somebody's asking, develop it properly. I'm curious about developing something fit for human use. That is way harder, and I can't. It's not something I can do in 2 hours. It's something I'd have to like spend a lot more time researching, which we don't have the time for that shit. Okay, so Isaac asks, wouldn't the effect of this gene in the inner ear diminish over time as the cells are replaced? No. This should also affect the stem cells in the area. And also, this isn't... so with the lactose thing, it existed as DNA that was sort of just floating around, like, separately. And so as the cells divide and copy, that bit of DNA eventually doesn't get copied properly. Whereas what we're doing here, because it's a CRISPR thing, it is permanently modifying the actual genome of those cells, and so they are literally going to be different forever, in theory, is the idea. Fuck knows if this would actually work. This is the sort of thing that you design and then you actually test it and see if it works, but we're going with it for the time being. Okay, so I'm going to set this to Plasmid. Wonderful. Okay, I'm going to close this because I want to be able to make this a little bigger. Okay, so here's our Plasmid again. You'll see that it looks a little bit different than when we first looked at it on AdGene because Benchlink shifts shit around a little bit. But the important part is here. So if we're looking at this, there was a note that said Kozak Sequence, if memory serves. What's this? EGFB. Vertebrate consensus sequence. Wow, that's annoying. Okay, we're gonna put that back. Okay. Okay, Tyler, if the gene stays in the cells forever and is copied, does that mean the genome of the entire body will change? No, because the idea with this is you're only modifying a small clump of cells. The stem cells that go on to form the rest of the body typically come from bone marrow. So the only way that this would affect everything is if you modified the bone marrow, which is extremely dangerous. The idea is we're trying to keep this confined. Because the cells don't actually divide very quickly in the inner ear. And sometimes they don't repair at all. This is why hearing loss is a thing. Whereas in other animals, if you like damage the hair cells, for example, they grow back. In humans, not so much. No one's really sure why, but there's some interesting studies on this. So, moving forward, basically what we can see. So we've got our SYN promoter here. It's Kar. And then this bit that I highlighted is called the Kozak sequence. And I'm not entirely sure that we actually need this. Because Kozak sequences are for... yeah, here we go. Okay, so this is where this would start, I suspect. I'm just doing a little checky poo here. Any weird stop codons? Nope, that's exactly in frame. Okay, cool. So, a Kozak sequence is actually this. I don't know if I've highlighted the entire thing or just most of it, but yeah, this label's a little bit off. So the Kozak sequence is sort of where a ribosome binds to get protein translation started. But we don't care about protein translation because we're literally only using this as guide RNA, so I don't actually think that this matters. But I'm going to include it anyway because might as well. So what we're going to be doing is, or the thing I'm looking for right now is where could we theoretically cut, and assuming, like, keeping in mind that we're not actually doing this, so this isn't actually necessary, per se. But I'm trying to show how you would design this if you were actually going to do this. So what we're looking for are restriction sites, which are these little tags above the code are restriction sites. So these are the names of specific enzymes which you can buy, that if you mix them with this bit of DNA, they will cut exactly right here. Now, we can't use this like CRISPR in a body, because this site will show up literally hundreds of times throughout the genome. So if you were to apply it to like human DNA, it would cut it to shreds and you're fucked. Whereas CRISPR will only cut once in theory. So when you're handling DNA, these are fine. Otherwise, not so much. So first things first, BAM-HI is the one that I'm thinking of using. So we open up this window, go to BAM-HI. It's already highlighted because I was using it for another project. But you can see that there's only one cut site, which is really, really important. If there was two, it would make two cuts in here, and then you'd end up with a chunk floating around that is not supposed to detach. So we're already doing good. We've got BAM-HI, and there's only one cut site. Perfect. So now let's scroll through. So basically what I want to do is I want to cut all of this crap out. It's not necessary. We don't need it. It's in the way. It's got to go. So we click on this piece, and then we scroll to the end of it. And we see that there's another restriction site right here. And you'll see that there's an ITR over here. So if we can cut out, if we were to cut with this and BAM-HI, in theory, it would just cut out all this crap, and then we could just stick our guide RNA sequence in here, and, you know, we're off to the fucking races, which would be great. So let's see. Okay, now we've got a problem. So you'll see that this one makes two cuts. Two cuts are bad. We don't want two cuts. So let's look at one of these other restriction sites that are over here. I like to use, when I'm designing stuff, I tend to gravitate to restriction enzymes I already own, which is just good practice, because then you don't need to buy another one because they're expensive. So I own both of these. I own NotI and EAGI. So let's see if either of them, see, that's a problem. EAGI has two sites, NotI has 2 sites. That's a problem. We don't want that. How about, so now we're into kind of frustrating territory. We might leave this poly A here, and maybe we'll cut a little higher and see. Oh, hey, that's where the other X-Hoy is. That actually works out really conveniently. Okay, so let's look at this again. So this was the one that I wanted to use originally because it cuts here. And I was worried because it said there were 2 cut sites, except when I click on the other cut site, it's here. So if we cut with X-Hoy, it's actually going to cut out all this stuff, which is actually quite helpful. So because it cuts twice, it cuts here, and it cuts here, it's as if it only cuts here. And the reason being, so I'll show it to you on Plasmid view. So it's showing 2 cut sites. It's showing a cut site here and a cut site here. So if we cut with BAM-HI, we get 1 cut here. If we cut with X-Hoy, we get 1 cut here, and 1 cut here. So let's actually do this digestion just so I can show you what happens. So I'm in the digestion menu. I've set it to. I've selected BAM-HI, which is what should cut at the start of this gene, and X-Hoy, which should cut at the end. And now we're going to run the digest. And so you'll see that 3 fragments get made, and so when I run a virtual digest, this will show you what I mean. Somebody just kicked in. Oh, Peter Schneider, thank you very much. Asked, can you make a video about plant cell culture? Yes, I'm already working on it. It's just, we'd already done a whole bunch of work, and then pandemic happened, and it killed all of my cultures. So, because I had to leave them alone for a month, so I've got to start that again. But I have all the stuff for it already. Okay, so basically, when you make these 2 cuts, you end up with 3 pieces of DNA, right? So, because you're getting a cut here, and a cut here. So you'd expect this one to be of length about 477. You'd expect this one, which goes from here to here, to be about 1390, about 1340. And then you'd expect the other one to be the rest, basically. Like you'd expect it to go from there to oh shit, okay. You'd expect it to be like this piece. So when we look at the virtual digest, that's exactly what we see. We see one piece, which is 477, right? We see another piece, which is the other chunk, and then we see this. In fact, I'm going to click it, and then we're going to go back to Plasmid view, and it should, see? So when you make those 2 cuts, all this crap gets cut out, and you end up with this piece right here, which is what we want. Because now we can put something new in this chunk, and then we're good to go. So I'm going to do exactly that. I'm going to go from this to... I'm going to just scroll up so that it doesn't shit the bed when I do this. So this is the chunk we want to cut out. We've already confirmed with the digest that it will do that. So before I do this, I'm going to go back to projects. This is 58867. Cool. I'm going to just make a duplicate of this because I don't want to, like, I want to make sure that any edits are non-destructive. This is really, really important. If you're going to make edits on a piece of DNA that you've got in your Benchlink thing, always make copies so you're not making destructive edits. Because if you make a mistake, it's very difficult to go back and undo things. Okay, cool. So we've got the thing highlighted that we want, and I'm literally just going to go delete, and it should go away. Or it'll shit the bed. We'll see. Delete. Go away. Yay, and it's gone. Okay. Wait. No, that's not right. It's not what we want. I need to double check this. Okay, so when we cut with BAM-HI, yeah, that's the problem. Okay, so I'm going to actually cut from here, because I want to leave this BAM-HI here so that we can put it back, because that's how this would work. Unless we did Gibson. Could do Gibson. Then we can just ignore this. I need to see the exploit site. Sorry, I'm mostly just thinking out loud because I just need to see how this works. Actually, no, that could work. If we cut from here. Okay, so let me explain my thinking here. There's several different ways to put the DNA back together once you've done this cut. Method number one is what's called golden gate assembly. So basically, you cut with these restriction enzymes, and then you design the piece of DNA that's going to go into that hole to also have the same restriction enzymes. So like the new piece would have, so this is what's called sticky. So you can see that it's this like S-shaped cut. So it leaves a bit of DNA that's dangling off the end, so that if you were to apply a matching piece of DNA, it'll literally go like boop and stick, and then you can reconnect it permanently. The other way to do this is with something called Gibson assembly, which is more complicated, and I'm not going to get into it at the moment. But what it does is it actually chews back this top side. So like literally like these letters get yeeted, and only the ones on the bottom stay. So it would start here. So basically it blunts the connection a little bit. So you lose a couple of letters, but it means that you can ignore the presence of these enzymes. So because this one has matching overhangs, we could do that. So I think we're gonna pretend that we're using Gibson. I don't know if we actually would, because again, we're not actually making this, but we're going to pretend that we are. So Gibson always chews back what's called the 5' end, which in this case is the bottom, and on the other side would be the top. So in this case, we'd lose till this C. So let me just check this again, just to make sure I'm not about to fuck this up. I am about to fuck this up because I grabbed it the wrong spot. Okay, well, shit. Fuck. I meant to grab this one. Okay, so, cuts here. Okay, so we're looking at here. In this case, so for Golden Gate, you would assume that the cut happens on this top edge. For Gibson, you assume the cut happens on the bottom edge, with this sort of geometry. So I'm grabbing from there. That's going to get blunted. That's fine. Now we scroll all the way back up, and we find this thing. Great. And we select there. Okay, now we've properly selected the chunk that we want to remove, and are going to cut properly so that it's... Basically, you've got to make sure that your design, when you're doing this, has to match the reality of when you go to do this in a lab. So it always helps to just double check things, because if you rush through this, you're going to fuck it up. So I've definitely selected the right stuff. So for real this time, I'm going to click delete. And you'll notice that there's no restriction sites here anymore. So I'm going to just make a little mark here. Little annotation. Just so that I know. Because as soon as I click off this, it's going to be just very difficult to find. So I'm going to make this a brighter color just so that I don't lose it, because that gray is hard to see. So I'm doing this to make sure that whatever we stick back in here is matching. Okay, I'm gonna just check the comments for a second because we've had some questions and some shit posting, so I need to address both. What the fuck? Okay, bye. Okay, so... yeah, I do want some mods. I have some people who normally mod, but they were busy today. So, shit happens. Okay, so we've got our cut site. Now the important, now the actual hard part. um, um, this was the easy bit, frankly. So also the other thing that I failed to mention is that we would also have to apply an extra piece of DNA which could not be delivered virally. So the thing with CRISPR is all CRISPR does is make a cut. If you want to put new DNA in the hole, you actually have to supply that DNA as a double-stranded linear segment of DNA. So this is why I think that doing the local delivery of the thing would work best, because then you would not only control which cells are getting cut, because we're using CRISPR on a neuron promoter, so it's only going to even be active in the right cell type. Because we're delivering only the DNA that we want to stick back in the hole at the cut site or at the injection site, it'll mean that any cells that accidentally get modified when they weren't supposed to will not have anything happen to them. The hole will just get patched back up, and then that should be fine. The other option, rather than a neuron-specific promoter, is a neuron-specific promoter and or a inducible promoter. So something that like turns on when you apply an antibiotic, for example. So there's a system that uses tetracycline as the inducible trigger, or doxycycline, or there's a few others. But the point is, you could basically apply the virus and make sure the code will only run when the antibiotic or other trigger is present. And so that way you could, modify the cells, apply the trigger briefly. CRISPR, like the Cas protein, will be produced. It'll make the cuts. The recombination event would happen where it sticks the new DNA in the hole. And then when you remove the antibiotic trigger, it's not going to keep trying to cut. Because the problem with CRISPR is especially Cas9, is it has a propensity to cut off target. So if you leave it running, it will essentially, and this is what I meant by like this is why I wouldn't do it this way if I was actually going to try this on humans. It has a propensity to cut the wrong thing. Even though we're going to be providing it with a guide, there's only 4 DNA letters. So there's only so many combinations of individual letters. So you will end up with off targets. And also sometimes it just doesn't stick to the right thing. So there's definitely some more precautions if I was going to actually use this on humans, but for the sake of today's video and discussion, we're going to just kind of breeze by that and pretend that's not a problem, even though it definitely is. Okay, so now we come to the bit where I actually need your help, because I was not able to find this on my own. So I can't pronounce your name. Megadon. There's Cas proteins with very high accuracy, though. I remember reading about one with no detectable off-target activity. Yes, but we're not using that one because I couldn't find that plasmid. Cas12A, I think, is much more accurate, but everybody uses Cas9 because that's what's available. It's not the best, it's just what's there. We're just doing this as a demonstration today. We're not doing this as if I was a pharmaceutical company and looking to actually make people frickin' detect magnets. So we're just go with it. Yeah, these ethical concerns are helpfully entirely theoretical, because you wouldn't actually do it this way. Like, to if I was it look if I was going to sit down and design this as if it was actually going to be used on humans, it would take me days. Like, it's not something I can do in 2 hours on a stream. So keeping that in mind, we're just going with what I'm going to call a minimum viable version, the sort of thing that you could like test on a mouse, just to prove that the system works before you go back in and make it better, right? Like, this is the sort of thing that you do it once. Because I mean, if you were to do this to a mouse using using the exact thing that we're going to make today, or almost the exact, you'd have to tweak it to be mouse. Then you could test it, prove that it would give the mouse the ability to detect magnetic fields, and then once you've done that, you could then, in a follow-up study, refine the constructs to be actually human usable. So, this isn't far off, but it's not perfect either. So yeah, this is the process. If you're doing a biological project like this, you always do minimum viable and test it on a mouse before you make it more complicated. Because if you try and do the more complicated thing, every extra bit that you add is a spot where something can go wrong. So maybe the modification itself could work, but maybe the tetracycline inducible thing isn't working properly in that tissue for whatever fucking reason. Like, there's a lot of different ways that things can go wrong. So you do minimum viable, prove that the basic idea works, and then you build on it. You don't try and you don't try and build an aircraft carrier before you know how to make a little like dinghy. Okay. So the page that I have open right now is actually the calcium voltage gated channel that we want. Except this one's from pigeons. So this one, and I'll explain what we're looking at in a second, but this is the version from pigeons, which should have the extra bit of code that allows them to detect the magnetic fields. So what we're looking at here, you'll see that there's this green line with a bunch of little like dash marks on it. I'm gonna zoom this out. Oh, that's fascinating. Okay, so this is the gene that we care about. So these lines, these horizontal line or vertical lines, are what are called exons. So in a mammalian gene, and this is mostly prevalent in animal, insect-ish genes. It's less present in bacteria, but it is possible. There are what are called introns and exons. So exons are the bits that actually get spliced together into the final thing that gets expressed. The introns are these chunks of code in between, which we don't know what they do. So there's literally hundreds and potentially thousands of nucleotides there. We just don't know what they do. Sometimes, depending on the protein. So like there's a protein for muscle, like a very specific type of muscle. Depending on which one of the exons that tissue type uses. So like, for example, in this type of muscle, like in, let's say, smooth muscle, it might use this exon, these ones, and these ones, but it might like ignore a couple of these. You would end up with one type of protein, whereas in another tissue type, it might use a different combination of exons, and you would end up with a similar but different protein, all coded for by the same gene. And this is very weird, but it is the way it is. And then there's the introns, which we don't really know what they do, and a lot of the time they're regulatory elements that like are expressed as RNA, but they don't necessarily make protein, but then the RNA that's left kind of like fucks off to another part of the cell and does like regulatory stuff, or maybe has catalytic activity. It's very weird. Biology doesn't make any sense. Like when I say that things are held together by zip ties and duct tape, this is the sort of shit that I'm talking about. Like, why would you code it this way? Like if humans were coding this, they would not do this way. They would just take all the important bits, shove them all together into one little concise chunk and be like, gene. Rather than biology, which is just like this nonsense where there's just shit everywhere and stuff in the middle, which we don't know what it does. So the question is, how do you even design around this complicated stuff carefully and through a lot of failure? Okay, so one of the things that this paper noted was the important bit. This exists in exon number 28. So if we go back to this page and scroll up a little bit, it'll say exon count 46. So in this clusterfuck of stuff, there are 46 exons. I pulled up the supplementary data, so this is not in the main paper. This is like you go to the main paper, and then you scroll all the way to the bottom, and then it gives you a link to the supplementary data, and you go to that link, and then you find this. So in the supplementary data, it gives you a map of the gene that we care about. This top one is the one that we give a shit about, and exon number 28, B, is the one that we care about. So we can see that this same pattern should show up on this gene, because I think it's the same species, but I could be wrong. Shit. Too much zoom. Zoom that out a little. Okay, so immediately we already know that we have a problem, because this does not look like the same thing. Does this scroll when I pull it? It does. Exciting. Actually, no, it does. It's just the graphic doesn't quite look right. God, this is just a fucking clusterfuck of stuff. Interesting. So the problem with this is there... Oh, come on. Again? Sorry, I gotta fix my light again. I'm gonna use more tape. Stick. Jesus. Next time I'm getting the fucking super glue. God damn. Okay, we're back. Alright. Yeah. I gotta get a proper soft box. Okay. Do I not have a mount? No, I have a piece of paper towel draped over the light to act as a softbox, because I never actually bought a proper softbox, because I'm a fucking idiot. Okay, so now here's the issue. This is... I need to know exactly what species this is from, because this doesn't actually say. And this is where... Okay, so this is where we run into our first problem. Because this is like, yeah, this is the layout of the exons, except of what fucking species? Like, if I don't know what species it is, how am I supposed to know? Like, this is going to vary wildly depending on the species. So, you know, if I don't have the right species, then this isn't going to look right, which it doesn't. Because, like, when I go here, like, I assumed it was rock pigeons, which is what this is from. Except this doesn't look right. This doesn't look right at all. It looks similar, but it doesn't look right. So, that's a problem. Considering this, I'm not entirely sure what to do. So let's just do a little searching around. Gene. Gene cards. Let's see what this is. Nope, I don't want health conditions. Okay, so I'm going to just be searching around a little bit, and this was, I kind of ran out of time while I was prepping for this stream. Otherwise, I'd have found this in advance. So, enhancer. Okay, we don't want that. We just want the gene sequence. Gene sequence. Okay, I'll take Uniprot. Fuck with Uniprot. Okay, so this is the human one. So now if we scroll down, yep, okay. Okay, so let's go back over to here, scroll up a little bit, And I'm going to actually use this as my guide here. LKL, oh, come on. You've got to be fucking kidding me. Okay, there's nothing more annoying than when people have text as an image in a paper. Because as you can see, I can't fucking highlight anything. So now I gotta type this out like a pleb. Alright, LKL VIA FKPK. Copy. Okay, I'm gonna go back to that Uniprot page. So just as a reminder for people who maybe are just tuning in for the same first time, Uniprot is a protein database. So we've already found. So if I scroll up, this is the protein. Like this is it, or at least this is the first subunit of this protein. So we've already found the thing. And what now that I'm thinking about this, just because of how difficult this is going to be to find the right reference, I might do this differently, and I might just say fuck it, let's just express this protein properly, rather than trying to mess with CRISPR, because targeting this might be hard if the protein hasn't been sequenced properly, or if the sequence is, let's go with, tedious to process. So let's see if this shows up. Okay, it should be around, because it gave a fucking reference to it. Okay, human here, 1254. 1254. Okay, so 12. Oh, for what the fuck? Okay, well, thank you, computer. That was literally the exact opposite thing that I wanted you to do, but you know. An attempt was made, so I'm just going to scroll up for a second. Okay, yeah, so the numbers are on the top, which is good. Need to know that. So 1254, numbers are on the top. So, wait, was it 1254 or 1245? No, it's 1254. Weird. Okay. So 1254 is here, so that's 12550. This is 1254. LKVIFK, yep. LK. KPK. This is already different. This is LKV IA. This is missing an L. That's super weird. That is super, super weird. But either way, KPK, this is where it ends. So this is the start part of the protein from this paper. So you see LKL, that L shouldn't be there. Oh! I misread it. That was totally my bad. That was totally my bad. LKVIA, so instead of LKL, I was looking for LKL, it's LKV, which I think was actually what was in here. Yeah, LKV, IAF, KPK. So this right here is the thing from the paper, which is correct, which is good. And then there's a gap, and then GYFS, blah, 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 blah. So if we go back over to here, it should go, there should be there is a gap here, which is to say that the chunk that we want to add goes right here, and then goes GYFS, blah, 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 blah. So all we have to do is essentially stick the correct chunk, which in this case, we're going to go with rock pigeon, is IFV RKKERWL. Keeping in mind that these letters are representative of amino acids. So this is the chunk that needs to go in there. See Ben. Has 2 amino acids different in the paper. LKVIF KPK. Does it? No, it doesn't. LKVIAF KPK. It's the same. Is my audio desynced? No, my audio is fine. It scared the crap out of me for a second there. Okay, anyways, so yeah, we've got to stick the correct thing here. So when I was first planning this out, I figured it would be easier. I didn't think it'd be easier. I thought it'd be more entertaining to use CRISPR. But the problem is I hadn't accounted for the difficulty of finding this sequence in uh, just finding the sequence, period, in the human genome. So targeting it would be a little tricky. So let's see what we have in the way of stuff here. Isoforms, yeah, that's not helpful. I want just DNA. Where would I find... Yeah, UniProt is really great when you want protein stuff, but it's absolute shite when you want the gene sequence that it came from. Gene cards? No, that's not right. XProt? No, that's not right. Okay, so... Audio's fine, audio's fine, no, it's fine, everything's fine. Okay. So originally my plan was, okay, let's just use this AAV thing and we will deliver CRISPR and a guide thing, and that's one way to do this. I think what would be easier, and this isn't ideal, but it would be easier, and I think what we might go for, because it's already been an hour and a half and I'm tired, and I've been talking up a storm. Yeah, we might save CRISPR for something a little easier, where the gene sequence is actually available, rather than an absolute pain in the dick to find. So what we might do is grab the FASTA sequence. Okay, so this is the actual, this is the entire protein sequence. So what we could do is grab this, figure out, but before we reverse translate it back into DNA, and then, so what we could do is we could take this, we could go over to Benchlink, and we could, actually, so if we go over to here, if we left some of this stuff in place, we could basically, actually, we can just use Bama Chat, we can make this as a fusion. So if we basically just, cut out just the GFP, we left all this other stuff in here, and then we stuck the protein that we want, like the calcium channel protein, UCSC genome browser. Wait. UCSC, okay. Only two of them. Where's the other one? Is it? Oh god. Yeah, technically that's the thing I want, I think, but that is such a mess that I don't even know what to do with this. What does that do? No, that's not better. Yeah, this is a fucking mess. I hate working with genomic stuff. What? Okay, so we're gonna do this two ways, I think. I'm gonna go back because this is not what I want. Go back to UniProt, you fuck. There we go. Okay, so what I'm gonna do is I'm gonna start working on this as if we're just gonna apply a new copy of the gene, because that's way easier and I don't need to deal with whatever the fuck that site was. However, if somebody can find me the exact gene sequence for this particular spot, so we just found it. Where the fuck did it go? It was, yeah, this. So if somebody can find me this part of the gene sequence, this is the amino sequence, so if somebody can basically back translate, figure out where the hell this is in the genome, we'll do CRISPR. I need to find I need to be staring at the exact spot in the genome where the CRISPR cut has to happen. If somebody can find me that code and post it in the chat, I'm going to keep an eye on the chat, we'll do CRISPR. Otherwise, we're going to do this the easy way because this shit's ridiculous. So, yeah, that's the plan. Because I don't know what the fuck that thing was I just opened, but it was a mess. So, God, I hate working with genomic stuff. This is so... I love doing this method so much more than ever fucking with CRISPR. Messing with CRISPR means you need to have the exact gene sequence, which means looking at sequence data, which is always a goddamn mess, because, like I said, it's held together by zip ties and duct tape and 4 billion years worth of bullshit. So it's just awful. So yeah, chat, I believe in you, but good luck. Okay, I'll just copy this. Now we're gonna go to faster. Okay, we're gonna just copy this. Where can I put this? That is convenient and easy. Where I can just make this mod really quick. I'm gonna do it here. Amino acid sequence. What is the name of this protein? CAC NA1D. D. Human. Create. Okay. We're going to paste that in. We're going to go Control F. We're going to go back. Oh, come on. It's going to make me fucking scroll again. Alright. So we're going to grab this again just as our marker, just so we get to the right area. Copy. Okay. Now we're going to go back to here. We're going to go Control F. Paste. Cool. This is going to get us to the right spot. KPK right here. Wonderful. Okay, I can close this. Okay, so let me just explain what I just did. We went to UniProt. I copied the entire gene sequence, or rather the entire amino sequence. We then stuck it into Benchlink as an amino acid, just separately, not stuck into the main file yet. We then found the spot where the extra couple of letters need to go. Now I'm gonna just write this out so that I have it. We're gonna use the rock pigeon one because pigeon, they're not sharks. Seems like it's appropriate. So IVFRKKERWL should be the sequence. Copy. So now this needs to go here. Pasty paste. Annotation. Pigeon insert. Okay. Okay, I'm gonna just have a look at the comments, see if anyone's come to the rescue. I feel like no. Okay. So we now have the corrected amino acid sequence for the protein, which, if expressed in the correct tissue type, should induce this magnetoreception thing. Very cool. Why is this still highlighted? That should not be highlighted. I cleared that, didn't I? Go away. Okay, cool. That was annoying. Don't know why I did that. We've now got the pigeon insert noted. So this is, it's kind of wild when you think about it. This is the only difference between being able to, theoretically, being able to detect magnetic fields and not. It's such a tiny little insert, but this is the weird shit with biology. The tiniest little change like this can have a huge, huge, huge effect. I'm gonna just reply to Super Ackman really quickly. Idea for a project: fix the messy code of an organism. You can't. It's not code you can fix. It's not, because we don't understand what it does. We don't know what most of it does. It's there. We know that if we fuck with it, shit breaks, but we don't know why. We don't know how, and it can break in 1,000 different ways. You can't make like nice, clean. You can't make a human with nice, clean code. It doesn't work like that, because that would literally require starting from basics, which we don't understand even close to enough about biology to be able to do that. It's just not a thing. What the fuck? So yeah. It's possible the insert wouldn't work the way we'd like it to in a human receptor, but I doubt it. The fact that it's so similar across all of the species that, or not all, but many of the species that are magnetoreceptive suggests to me that it's pretty conserved and it's pretty consistent. Yeah, junk DNA is not a thing. We now call it dark DNA, not junk. Anyone who calls it junk DNA is a fucking idiot. It's dark DNA because we don't know what it does. Also, a lot of the like junk DNA is only junk in the sense that it's not running in healthy individuals, which is what we tend to study. However, when you study sick individuals, the DNA lights up like a fucking Christmas tree because most of it's there to deal with niche cases. It's not there to deal with your like everyday, the biology is fine. It's there with like a pathogen is now infected and a whole bunch of genes turn on that we didn't think were there to actually deal with this pathogen or problem or whatever the case may be. So dark DNA is the more accurate modern term, not junk DNA. It's not junk. It does a job. We just don't know what it is. Okay. So once you were to add this to a recipient, they'd probably feel pretty weird for a while. Like, they'd probably be really disoriented for like a week or two, but then the brain would adjust. Like, it'd take you a little while to figure out what that is, but it'd figure it out. Like, in the same way that, like, you know, if you're easily seasick, the first couple of days on a boat are not going to be fun. But after a month of being on a boat, like, it doesn't matter how seasick you were before, you're fine. You get your sea legs, and then you're just used to it. Your brain edits out the stimulus or just learns to adjust to it. So with this, if all of a sudden you're getting this new stimulus of like, what is that feeling? I feel like I'm queasy all the time. Like it feels like the whole world's shifting around. You'd get used to it, and then eventually you'd learn to interpret, oh, okay, that's the magnetic field. Like, when it feels weird to look this way but not that way, that feeling is, oh, that's north, or whatever the case may be. Oh my god, did somebody actually find it? Okay, we're gonna check this. Okay, we're gonna check this out really quick, and then we're gonna see if this is legit. Okay, sequence ID. Sequence ID. XM 01153 4097.2 4097.2. Okay, I'll bite. Where did you find this? Can you Carnage spelled with 2 K's. Can you just post a link to that? Where have you just found that? So I don't have to dig through a bunch of random shit. What is this? Oh, yeah, no, okay. That's a thing. You can post links, but YouTube will automatically hide them, but I have the option to let them through. I mean, or just post what site you found that on, because I mean, this looks right. I can't find it. Clinical significance, NCBI. I'd really just like a frickin'... Let's just lose the sequence ID, see if that shows up. No, it gives me the same thing. Allele registry, interfering RNA registry, BioGPS. That's a new site. Never been on this. Oh God, what is this? No, not what I want. Oh, Vivek. See, when Vivek talks, I listen. EU 36. How many threes is that? 333 on NCBI. Homo sapiens, calcium channel voltage dependent, mRNA. Okay, so the issue with mRNA is this has already been parsed down. I really do need the gene sequence, because with this, I mean this is basically what's on UniProt, which is good in theory, but wait a second, how long is this? Yeah, no. So yeah, this is the mRNA. So this is basically direct 1-to-1 what ends up coming out without the actual exon sequence. I need the actual genomic sequence, because CRISPR doesn't work on mRNA, because this code is not what's going to be present in the genome. This is what shows up after you removed all the introns. So yeah, this is close, but I really need the gene that this came from. Session number. This is really cool. Okay, I'm gonna keep working on the other thing. You guys keep looking. Yeah, so I need the actual DNA, not the mRNA. I need this, but as it appears in the genome, so that way we can cut the genome in the right spot. But okay, so considering that we're still waiting on that, we're going to move forward on this other method, because this other method is basically done. So copy. Back translate. That's what I want to see. Select organism. Homo sapien. Okay, that's fine. I don't care about this. Actually, yeah, avoid hairpins. Preview. It's thinking. Oh. Save as a new sequence. Yep, that's fine. It should do the thing. What did it... Yay! Okay, cool. It just did the thing. Wonderful. Okay. Control A. We're going to just annotate this so that it's already annotated when we stick it in the next thing. Okay, annotation name. It's CACNA1D. Human. Plus pigeon. It's going to be pink, because why not? Okay, copy. That should copy the entire thing. Yes, it did. Wonderful. Great. We're going to go to here. This should be this. I'm going to make another copy of this, because, remember, non-destructive edits. What the fuck is going on? Okay. We've got our copy of the copy. Cool, so now I can actually do this. We're going to use NCOI this time instead of MHI, so I'm going to just adjust this. Just make sure this works. NCOI, yes, wonderful. Lose MHI. Now I just want to cut at the end of this GFP because I don't want to mess with any other stuff that's in here. Oh, E-core I. Perfect. That's exactly what I like to see. I'm just burning through this real quick. Oh, yeah. That's what I like to see. It fucking works. Okay, great. Okie dokie. Before I do anything else, let me just quickly review what I just did, which is almost nothing. But, okay. So in this plasmid, right? I'm going to close this so I can make this bigger. In this plasmid, we've got our promoter here that is neuron specific. So this should be this bit of DNA will only run when it's in a neuron. The ITRs should be in here. Yeah. This is AAV based, so this is good. Excuse me. What? Okay. Man, if you've got Fuser questions, go to fuser.net. There's literally an entire website to answer your questions. Okay, moving on. What's Joshua saying? Okay, you don't understand. That's fine. Don't care. Moving on. Okay, so this is our neuron-specific promoter. This is GFP, which we want to get rid of. In theory, we could leave it in there. Haven't decided if I want to do that or not, but I'm thinking we're going to remove it. So the way that we're going to remove it is we're going to use two restriction enzymes, this NCOI and this other one, which is ECORI. So these cut right at the start and end of this sequence. So if we cut it out, it should leave a nice gap here. So that way we can stick this into the thing. And I'm going to also create translation just so that it's consistent. So before we paste this in, I'm just going to make sure that it has this. So we're going to just pretend we're doing Golden Gate here. Because it's convenient and it's easy and it's fast. Okie dokie. So just click this, should bring it back up to the top. Okay. So NCOI goes from this C to this G. So we're just going to copy that whole chunk. This could be a problem. I should use BAM-HI. I should really use BAM-HI. Yeah, fuck it. I'm gonna use BAM-HI instead. It's gonna be less destructive. And then I can just include the thing. Okay, so the reason I'm changing which enzyme I'm gonna whatever. Okay, so let me just show you something really quickly. So NCOI, you can see that it cuts here. The one problem is one of the letters that it wants is this G right here on the end, which is the start of the next. So there's GFP starts here. It starts with an M, then it goes to a V. The problem is when you look at the translation of the thing that we want to stick in here, it goes from an M to another M. So these letters do not match, which is a problem because if we tried to use this restriction site, we'd have to change one of the letters here. So we'd have to change this A to be a G, which would change what amino acid this is, which could fuck with the sequence. I don't know if it matters, but it might. So considering that, we're going to use a different one and then just stick stuff back in, basically. So we're going to go back over here. BAM-HI, so I need the whole BAM-HI site. So I'm going to ignore this G and we're going to pretend that it would just be here. Fuck it, I'll stick it in, why not? Okay, where does this go to? It stops right there. Great. And I'm going to actually keep copying all the way to this first M because I think it's important. So we're going to grab this whole thing. We're going to stick it right at the front of this. I'm going to do this and then I'm going to have a look at the comments, see if anyone's been super helpful. So, okay, the reason that I just did this is because now you could basically make, and I'm going to have to do the same thing on the back end in a second, but the idea is you could print this huge chunk of DNA, which would be just ungodly expensive, but doable. You could print this whole thing, and then when you want to stick this insert into this plasmid backbone, you could cut with these two enzymes, purify out the linear fragment that it gives you, cut the insert with the same two enzymes, and then just mix them, and they'll stick. Like, they'll stick together, you'll end up with a perfect plasmid, no big deal, end of problem. So, considering that, I also included all of the stuff at the beginning here because of the Kozak sequence, which I mentioned earlier, which is the spot where ribosomes actually bind. So I wanted to make sure that I preserve that in case that, because this is important. Like, if it's not here, the gene won't be expressed properly. So I'm just leaving it in there so that it's preserved, so I don't break anything. So yeah. I'm going to just take a quick little peek in the comments, see if somebody has come up with the fuck? Yeah, so just in case anyone's come up with the solution to this, your question from earlier. What was your question from earlier? Oh, you asked about the yeast thing. Okay, remind me when we get finished with this, and then I'll talk about how you do the yeast thing. Okay. So yeah, we're just trying to preserve stuff. I don't like breaking things if I don't have to, because in theory, this piece of DNA has been tested and works. Like, I saw a picture of it being used in a paper, so in theory it works. So the less we fuck with it, the better, whilst still adding the new functionality that we want. So cool. Yes, wonderful, lovely. So now we're going to go to the back end, which is about here. So EcoRI. So now this, I need to be a lot less picky about. So I'm going to just grab to there, because I don't know if this has a stopcode on. So we're going to look all the way to the bottom of this thing. Yes, okay, this was my suspicion. Okay, so now we're looking at the... Fuck, there's a BAM-HI site there. That's a problem. I should have checked that in advance, and I didn't because I'm stupid. Oh, there's 3 of them. Fuck. Son of a bitch. And there's NCOI. That's a problem. That's a problem. Fuck, I'd have to make a spacer. Shit. Okay, I just caught a problem. So we're gonna sort this in just like 2 seconds. Okay, BAM-HI. So I added this one, but unfortunately there's already 2 BAM-HI sites in this gene. So if we were to cut it with BAM-HI, it would just cut it to ribbons, which is not helpful. EcoRI is even worse. There's 4 of them. So we can't actually use either of these. So what we're going to do is we're going to just go through the restriction enzyme list, and we're going to make a couple little changes. And we're going to fix this, because that's fucking annoying. So okay, we're going to just get rid of this, and we're going to fix it, and then we're going to be done. And that'll be that. Okay. So let's just look at the list here. I'm just scanning through... Ooh, AGI. I like AGI. AGI is good. We're going to use AGI, and we're going to use... Do I have anything else I have? Okay, so I'm just going through this list looking for enzymes that have 0 cuts, because we want to just make sure that it's not going to cut in the wrong spot on the plasmid, because that would be a problem. Bunch of like weird shit, but nothing common. Ooh, Hin 3. I have Hin 3. Okay. Okay, we're gonna make a little spacer. Okay, exciting. Very exciting. Well, now it's not gonna conserve the Kozak sequence, which is a bit very annoying. Okay. Okay, sorry, I'm gonna be quiet for a second because I'm just thinking how the hell I fix this. Ooh, there's hidden 3 right there. That's helpful. Okay, let's just see how this looks on this side. I wanted to use AGI, which has 0 cuts, which is good, and hidden 3, which is also good. Hidden 3 I don't need to add. Ecori I can ignore because I'm just not going to use it. I'm just going to use hidden 3. And that should sort most of my problem. I just need to add an AGI side at the front. Okay, that's doable. Okay, so we're going to make a little spacer. So, dus to dus, copy. Okay, paste. So we're going to stick that back in. But then I'm going to grab AGI, I'm going to grab its sequence. No, I just need the top line. Thank you. Copy. Come on. Paste, you fucker. So this is going to mess with the Kozak sequence. So I don't know how that's going to behave, but it is the way that it is. Okay, we didn't accidentally introduce an ATG. No, we did not. Wonderful. Okay, so this is now a spacer. So what you would do is you would get, come on, where's the thing? Yeah, so from here to here, you would basically get this printed separately. And in fact, you don't even actually need to cut with anything, because you could just get this top line. Where does this go to? To the A. So you could basically get this, like that whole, like the whole thing. You can get this made, right? You could then take your tube of this DNA, cut it with AGI, so that it exposes what's called this sticky end. So you get this like overhang here. So like when I highlight it, you can see that the CCGG has the line underneath it. That would be the dangly bit. That would be what's overhanging. So then what you could do is you could cut the other piece. So you would cut this, let's close this, with BAM-HI and HIN 3, which we'll talk about HIN 3 in a second. But you can cut this with BAM-HI and HIN 3, and then mix in this piece, the big piece of DNA that you want, and then have 2 little short pieces made, and they're real tiny. Like, I don't even know if people would make this for you. You might have to make it longer on purpose and then like cut it. So like what you could do, because this is actually too short. So yeah, this from here to here, that's too short. Actually, no, length 12. Yeah, but the underside though. Yeah. Okay, this is too short. So nobody would make this for you. Just because it's too short. So what you would do is you would just add a bunch of junk here. So, no. AGTC, I'm just going to, just a bunch of random crap. So basically what you would do is you would have this chunk made to here. And you would have it made in 2 pieces. So you would have this top part made exactly like this, but then the bottom part. Actually, I mean, the easiest way to do this is you just go to the back end of the AGI. So you go to, like, here. So if you were to have this piece made, right, what you could do is you could make these as individual single strands. So, like, a company that would sell you primers for PCR, you could order these 2 single strands of DNA, mix them in a tube, heat them up, cool them down, and they'll stick together. And so you'll end up with exactly this chunk, right? You could then take that piece of now double-stranded DNA in a tube, treat it with AGI and BAM-HI, which would give you 2 fragments. This junk crap on the front, which you can ignore, and then this part, which is, well, it'd be a little longer, but one more. So, and then this part, which is the actual spacer that you want, you could then take, and then of course you heat it up to kill all the enzymes to make sure that they're not a problem. You would then have, this, assume that I selected the whole thing. So, then you take this piece, which you'd had made separately, you treat it with AGI. So now you've got this sticky end on this piece, right? On this piece. You have your little spacer, which has a sticky end on both ends. And then you mix it with, excuse me, this, which would be pre-cut. So, you cut this plasmid, you cut your spacer, and you cut the actual insert. And then you mix them, and they should just stick together nicely, and it should solve this problem. The Kozak sequence got a little fucked, so that's not ideal, but it's also the only way I can think of doing this that wouldn't mess with the sequence of the enzyme. So, yeah, fuck knows if this would work. You'd have to try it and see what happens. Okay, so that solves the issue of what to do about the front. The back should be much easier. Where did the thing go? Okay, I need the back. I just need Hind 3. I mean, I can just do this by hand. There's no reason I need to do this by copying that. Okay, Hind 3. We're just going to copy this. Okay, so we're adding a Hind 3 site to the back end, because remember, you've got to cut twice. So I'm going to add two things at the same time here. So I'm going to add a stop codon, which basically is what the ribosome. So the thing that the protein that reads this, what's going to be RNA, and turns it into protein. When it hits the stop codon, that tells the thing that the protein is over and to let go, or at the very least stop making protein. So we're going to add that. So TAA, TAG, TAA. I like to add more than one just to make sure it's like stop, in case it misses one or something. And then we're also going to paste in this Hind 3 site. Paste that in. So now if I make a translate forward, okay. So you'll see that there's the Hind 3 site here, and then these are stop codons. They're the little stars. So now the idea is I should be able to grab from here. So this is the insert. This is the thing that you're paying a company to make for you. What's going on? So yeah, you would pay some company to make this gigantic insert for you, which would cost you an arm and a leg, but they'd do it if you pay them enough. In fact, you would probably make it so that this would be loaded into some other plasmid just for carrying, basically, and then you would cut it out and do the handling. But anyway, so now I can grab from Hind 3, so it's going to grab from the start here, and I go all the way up. And we're going to grab from the BAM-HI, because all this stuff is included, because we've made the spacers properly. We're going to copy it. Now we're going to go over to here. Now we're going to click, we're going to basically do the exact opposite. So we're going to go from BAM-HI. We're going to scroll down. Here's Hind 3. We're going to select it. We're going to hit delete. So that should get rid of all of that stuff. Where in the hell did it just put me? Okay, cool. And now we're going to paste in our old fragment, which should return those cut sites, which it did. I can now see it. There's Hind 3, there's BAM-HI. So now what this should do, we have made a plasmid which expresses only under neurons, so it'll only be produced in neurons. It's made via an AAV, so you can just deliver it via a virus, and it would express this modified CACNA1D protein, which if you were to inject this directly into the semicircular canal's base, where the clump of neurons that's responsible for the actual sensing mechanism, it should start expressing this protein as well as the original version, and it should make you more sensitive to electrical impulses, or electrical currents, rather, induced in the semicircular canal when magnetic fields are present, which should, in theory, make you magnetoreceptive. Holy crap, that was a lot to say. Okay. Oh. That was a lot of work. CBI. thing. Okay, Carnage might have found it, but okay. So this is our. We're gonna call this version 1. I'm gonna now rename this. This is copy of that gene. Blah 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 blah. Okay, rename. What's the actual name of this on Adgene? PAAV Sin. Okay. PAAV Sin with pigeonized human CAC NA1D. Yeah. Pigeonized. Favorite new word. Okay. It says link's not working in chat. Oh, right. You either need to be in motion, or the magnetic field needs to be changing. So it should be set off when you're near alternating electric fields, so like power lines, microwaves, that sort of thing, which is weird. Like you wouldn't hear it, you'd just feel weird when you're near a microwave, which is hilarious, or a power line, or any of that kind of shit. But also if you're traveling. So like if you're in a car, you'd be able to navigate better, which is weird. Okay, show that. So yeah, this we're gonna call this version 1, and we're probably I'm gonna just take a quick look at the thing that Carnage just found, because it might be the gene sequence. We might be able to do the cast thing really quickly, but this is already a functional-ish version of this. Like in theory, you could have this made. In fact, you could basically have a company just do all of this for you. Like you just give them instructions. You're like, print this, cut this, stick this here, do this there, and they'll just do all of it for you. It'll cost you a little more, but like, 6,000 base pairs is looking at around $2,500, plus all the other little bits and pieces and sticking it in different places and assembly. You're probably looking at anywhere from $2,500 to $3,000 to do this. Then you could have another company make the AAVs for you, which would cost you about another $1,000, anywhere from $800 to $1,000, depending on the amount that you want, which would get you enough to actually try this on a mouse, probably. So yeah. Okay, so let's just have a look at the thing that Carnage found. NG_03299.1. Homo sapien calcium voltage gated channel subunit alpha 1 on chromosome, please. Okay. Is this going to be gigantic? This is going to be gigantic. Oh, God. Yeah, I mean, it's in there. In theory. I just need to figure out where in the hell in this massive clusterfuck of stuff it actually is. Okay, this is closer. Okay, so this is referencing all the exons. Okay. Oh, shit. Number 2, number 3, okay, I can work with this. Yeah, okay, these are all the different exons. So we need exon 28, which is this. If I click this, what's this going to do? Is this going to jump to the... Oh, yes! That's handy. Okay. That's kind of what I'm looking for. Okay, I'm going to just copy this really quick, and I'm going to just poke around on Benchlink. So yeah, Carnage might have found the thing that we need. I'm going to do this. Exon 28. So let's just see what we get. Did it do the thing? Yeah, okay. Boop. Pasty paste. What? No. What the fuck? Copy. No. It's not copying properly. Oh, because it's not highlighted. Because I'm an idiot. Okay. Copy. Okay, is this going to shit the bed when I post this in here? Okay, yeah, it got rid of the extra stuff. Okay. So, forward. No, that's not right. No. Delete translation. I'm going to start from one letter over. Okay, so what I'm just doing right now is I'm just walking this forward until I find where this is in frame. Forward. Which... Okay, now I'm looking for the thing that we modified on the last one, which I'm not seeing. Where did that go? Oh god. Wait, okay. Yeah, here. Yeah, we're looking for KPK. Exon 28. I don't see KPK. Yeah, I don't think this is right. It could just be in a different exon, though. I mean, also, I could have just frame shifted this, and this could just be wrong. So I'm going to just grab it from another letter, and we're going to see if that fixes it. Okay, create... What? Create forward. No, that is not better. That is definitely not better. I was right the first time. Delete translation. Okay, so yeah, this is in frame. But I'm not seeing the chunk that we need. So I'm going to go back over here. Oh god, it's going to be fucking scroll up again. Oh, okay, that's actually really handy. I can just copy this and see what happens. Let's try exon 29, because this is a 28B, which is just, what the fuck, like, there's no A and B, so I don't know how the fuck that's supposed to work. Let's make a new DNA sequence. We're going to call this exon 29, because that's what it is. Create. Okay. Paste. It'll get rid of the numbers. Let's make a translation forward. My god, that's in frame. Okay. I don't see it. Yeah, I'm not seeing it. Crazy Uncle Bob, thank you for your donation. So, could I give myself magnetoreception? I mean, technically, it just might give you ear cancer. This is why you test on mice first. Yes, it was exon 28 in the pigeon genome, but it is evidently not exon 28 in the human genome. So this is exon 28. You'll notice there is no KPK. There's a lot of stuff, but no KPK. So that's a problem. Exon 29 is similarly useless. So rather than go through every single exon, I think we might just call it, because this is, I mean, we're close. I mean, I appreciate you finding this, but I don't know if it's in here. Actually, wait a second. I can figure out where this is. Okay, let me just write some of this out. KA, oh, come on. Fuck it, I'll just use the lower thing. SSPFEYMMF. Copy. Now if I go to this, which is all aminos, and I paste that in, I just figured out where we are. It's exon 30. Okay, so let me just explain what I just did. Or maybe it is exon 29. What the fuck? No, it's exon 30. Okay. Control F. Paste. Right. Okay. So this is exon 29. I grabbed literally just a random string of amino acids just to kind of get a sense of where we're at. So I said SSPFEYM. I then went to the amino acid sequence, which we know is the right thing, and SSPFEYM is right here. So we're close. We're very, very close. So now if I go back to here, I should be able to nip over one more exon, and where did it go? Okay. This might be too short. We'll see, but this might be too short. We'll see. Okay, let's make a new one. This is insane. This is the weirdest, most difficult DNA handling I've ever done. Exon 30. God, I wish people would just annotate their shit properly. Okay. Paste. Oh god. No. Okay. Translate. KPK. Oh my god, I found it. It's literally right at the end. Holy dicks, we actually found it. Oh my god. So it's got to be right at the start of the end. Well, okay, well, I could just stick it there. I could use exon 30. Holy shit, we found it. Dude, fucking Carnage, you are a legend. I don't even know what to do with myself. Okay. So this is it. We found it. We totally freaking found it. Okay, I'm going to just grab a chunk of this. Copy. And I just want to see where the next exon is, because I assume it's far away. 263521. Yeah, it's way down there. Okay, wait. That's still highlighted, so I can scroll. Yeah, there's this huge gap here. So this is all intron. All of this is just useless intron garbage. Whereas this is where the next set of letters start. GGG is glycine, which I remember from this. So if we go back over to here. Yeah, that's it. Holy shit, we actually found the spot. My god. Okay. Now I just don't know how the fuck we put this back in here. Do you stick it on the front edge, or do you stick it on the back edge? How do you make sure that it's still an intron? I have no idea. I haven't the slightest fucking clue how you actually do this. Because I don't know how you mess with introns. I've never messed with introns before. It could be the bit after. Okay, I'm going to delete exon 30. Or rather, I'm going to select the whole thing. I'm going to delete that. Paste in the bigger chunk. And then I'm going to go back over to here. Grab exon 30 again. So I just want exon 30. This is the only part that matters. Copy. So now we're going to go back over to Benchlink. We're going to go Control F. Find me this, please. Oh, come on. You fuck. Okay. We're going to paste it here. Then I get to do the really tedious bollocks of getting rid of the spaces so that this formats properly. Get rid of all this crap. Oh, no, not that. Okay. Sorry, this is just going to take a second. It's going to be really boring, but we're going to get it done. Copy. Escape. F. Ah, no. No. Yes. Paste. Huzzah, I found. Okay, we're going to annotate this. That's exon 30. Okay. Holy shit. I can't believe we actually found that. I was not expecting you guys to actually find that. Power of the internet. Yeah, seriously, 50 internet points to Carnage for finding that. I mean, it's in the correct frame. You can tell because the amino sequence is correct. Yeah, Kozak Mutant, you're right. That's the fact that we made it this far is already further than I thought we'd get today, if I'm being completely honest. Holy shit. Okay. Okay, so just to reiterate what I've done here. See, that's a GT. Okay, intron, exon. Okay. Intron splicing. I just need to see what the signal is, because this is like drawing on shit I haven't thought about in 8 years. Oh god. Okay, what are the letters? I don't remember. I don't remember any of this shit. How does it... Exon, 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 splicing. Come on. Oop, no, just exon. Okay, so there should be a couple of letters that mark the start and end of the exons. Fuck me if I remember them. Tag? Is that tag? God, I don't remember any of this shit. I had a freaking test on this when I was in university, but that was, God, 7 years ago. And I have not thought about this since. Everything between the exons are introns, so long as it's in frame, you're good to go. Well, yeah, but if I mess with the ends of the exon, which is what I'm going to have to do to fix this. Okay, so what I got to do, if this is the piece of DNA that we would have to target the CRISPR thing to, but I don't remember. I think this GTG is the tag that says this is the end of the exon. I think. I don't remember. I'm going off of memory here, and this is what I was trying to find really quickly. If this GTG marks the end of the exon, I should be able to stick whatever I want, theoretically, here, and it should be counted as part of the exon, and I should be able to add the extra amino acids that I want. No, that's not it. That's not one. It's this. This is the thing that I want. The pigeon area should be marked, in theory. And it's not, because of course it isn't. Okay, well that's fun. Balls. Yeah, okay. Need to grab this part. Copy. I'm just trying to find what the frickin' DNA sequence is. Wait, what am I doing? That's not right. I don't need that. I just need the pigeon part. Oh, that was why. Okay. Right, yes. Okay. I click copy. No, it's just going to copy it directly. Click back translate. Homo sapiens. I don't know what this just saved that as. That's modified just now. Okay, yeah, this is the pigeon one. Okay, so copy. What? No, don't just fucking copy. Copy. Okay. So now if we go back to exon 30, that thing I just copied needs to be stuck right here, and I'm gonna just do that, and we're gonna label it, and that's gonna be the thing. Come on, paste. Fuck. There we go. Annotation. Pigeon insert. Save. So essentially what we've got to do is we would have to modify the original genome, and I'm going to make a translation of this just to make sure that it all looks fine, which it does. We would have to cut the genome here and stick this piece in here and hope that it fixes it. Leave this for the next stream. It's too much. Yeah, I mean, I don't disagree. We've already been at this for a while. I'm just kind of thinking this through. I mean, we might do a part 2, but the thing is, I don't. I mean, I could read up on how to do the CRISPR targeting thing, and the other thing. How do I want to handle this? I do not know. G 5 prime splices. Yeah. Well, why is there an R in that? Why are there M and R's in that splice site? That's not helpful. Fuck yeah. The thing is, I don't want to. if we're gonna do this, I want to just get it done. But I'm tired and my voice hurts from talking constantly for 2.5 hours. But we finally found the thing, so like I kind of want to just get it done. Butts. Okay. Exon splice site sequence. See what that looks like. RNA splicing, introns, exons, and these splices on. It's gonna be a whole frickin' paper. I need a picture. I need a picture that gives me the 3 letters that tells me what the fuck this is, and then I can just move on. Self-splicing. Oh god. M is A or C, and R is A or G. The thing is, I don't know if we're close, because this could be actually a lot more work. So I think I might just call it, because we made a thing that'll work. So I think what I might do is I'll read up on this, and then at the start of the next stream, you know what I think we're gonna do? We're gonna do an easy one next time, something that's like quick, and we'll finish this off in the next one. So that way I can have time to read up on how you deal with the intron exon bollocks. Figure out just I need to read up more on the CRISPR targeting system, and because I know there's some peculiarities with the specific places where it can cut. So I'm gonna read up on that, and I'm gonna read up on the intron stuff, and then the next one I think we'll probably do like tearless onions or something, because that one should be really quick. And we'll finish this off in the next one, and I think that's probably the best way to handle this. So yeah. So we're just gonna do some questions for the time being, and we're gonna just let this rest, and we'll come back to this in the next stream. Okay, so not the next stream. The next stream is gonna be a biohack chat on Friday with Gabriel. That's gonna be fun. We're gonna be talking about permaculture. So if you've got permaculture questions, you can send them to me on Twitter. But next Tuesday, when we go to do the next one of the Who's Gene streams, we'll finish this up. So we'll finish this up, we'll do whatever the topic is. I'm going to make sure that the options that I put up on the poll are fairly easy and quick, so that way we can just like burn through it really quick and then finish this up. I also need to address the yeast question from earlier really quickly, and we're going to do, so yeah, we're going to do like maybe 10-15 minutes of question time, and then we're going to call it for the day, because this has already been crazy long and I'm freaking tired. Okay, so I don't remember who asked the question about the yeast thing, but I'm going to just answer it, and if you're not watching anymore, you can come back and find this part, and then your answer. Okay, so if you want to do multiple genes in yeast, there's 2 ways to do it, to do so. This is method number 1, which oh, come on, fucker, it's not going to even show it. For fuck's sake. Okay, so we're going to backtrack a little bit, and I'll show you the other method first. Milk and eggs. Casey. Okay, so, okay, good. Seth is still here. So there's 2 ways to do this. This is the way that I tend to do it. So basically this is 4 genes. It's not 8, but it's still a lot. So I've got my yeast promoter here, and then, gene number 1, and then at the end of the gene, I have this, what's called a 2A peptide. So it's this short little sequence that basically makes the ribosome skip like a record. So think of literally like a record, like going like, so it makes the ribosome let go of the first protein and then immediately start transcribing the next one. And so you can get multiple proteins off of 1 promoter, which is, I like it because it's efficient, but that it comes with its issues. It doesn't necessarily always cut properly. But yeah, so the way I've got this set up, and this is actually a plasmid that is currently being synthesized for me. Like, I've paid for it, like it's happening. So this one actually has, and I'm gonna zoom in a little bit. just so it's easier to see what's going on. So you'll see that there's beta casein, kappa casein, alpha lactobutmin, beta lactoglobulin. These are my 4 different proteins that I want, and they're all connected by these 2A peptides. There's multiple 2A peptides. There's a list of them available on AdGene. You just stick them between your thing, and you make sure that everything's all in frame. But as long as they're there, you'll get multiple proteins. The other way to do this is something I'm just going to look up really quickly on AdGene, because I can just show it and it's easier. And we're going to actually be talking about this in a video that's coming up that I'm editing currently. I mean, not currently, I'm streaming right now, but like the rest of my day is going to be editing this video. I need to go to the Plasmid page so I can show this. So the other way to do this is you just build a really big plasmid with multiple different promoters, or you can use the same promoter over and over again. It's just not going to work as well. So in this case, there's 4 genes, technically 5 because 2 of them are fused together. They each have a promoter and a terminator before and after, and you just chain them all together. I mean, you don't need to immediately chain them. Like, you can have spacers and stuff between them. But if you just want more genes, you just add more promoters and more terminators and more genes. And you just end up with a bigger plasmid. So this is a very, very, very large plasmid. Because there's a lot of stuff going on. But it's, yeast can handle it. Yeast can handle up to like 100,000 base pairs in a plasmid. Like, they can handle really big. The only problem is physically handling a piece of DNA that large is a problem because it tends to break literally in your pipette. So that becomes its own problem. But yeah, if you want to make a yeast thing with 8 genes in it, you can totally do that. Just lots of promoters and terminators. So yeah, hopefully that answers your question. Okay, Ben asks, hi, what should I choose to study? Biology, genetics, microbiology, or I'm going to assume you mean biophysics. It depends what you want to do. If there's a synthetic biology thing, or a molecular biology thing, that would be my suggestion. Otherwise, biochemistry will get you all of the skills you need to do this kind of stuff. If you study general biology, it tends to be more broad. And less specific, which is why I dropped out. Like, I took general biology and it was just boring as shit. So, I mean, it wasn't boring. It was just like I wasn't learning this stuff. So yeah, wasn't into it. Spencer Minette asks, I'm a software and machine learning engineer who'd love to help. Any more functionality for an open source browser extension with better pasting, etc. that you'd want? I have no idea. Yeah, I've got nothing. I honestly, in terms of browser extensions, I honestly can't think of anything that I would really want. I mean, that one that lets you copy text from images is pretty cool. I'm into that. If you can make that better, that would be good. But other than that, I honestly, I don't know. Ken asks, I'm 3rd year in uni and still crap at reading and understanding papers. What do you suggest I do to improve my skills? Read more papers. That's literally the best answer I can give you. Just like, read more papers. Just figure out what about papers is important. I mean, I tend to just mostly read the methods and results because that's the bits I care about. But just read more papers. Just find papers you find interesting and just read them. I like, your professors will probably give you really boring papers to read, but if you can find something you're interested in so that you can be engaged enough to enjoy reading it, that tends to be how your reading comprehension improves. Is it possible to create biometal? Like metal from biology? Yeah. There's some bacteria that do it. But it only works with very specific metals and that sort of thing. You can do biomineralization, which tend to give you oxides and that sort of thing. There's also plants that are hyperaccumulators of particular metal salts. So like there's one that produces or that accumulates so much nickel that like if you cut it, it bleeds like green blue goo, which is like mostly nickel salts. Pretty toxic if you get it on you because that shit's bad for you, but like it's fine for the plant. it'd be hard to actually turn that into nickel metal, though. I'm working on VR AI. Is there any way that VR could help out with gene editing? Other than fold it, no. Not as far as I know. It's not the right tool. Would auto-matching sequences with NCBI or Uniprot add much? Yeah, I mean, if there was a better way of parsing NCBI to, like, so, like, if there was a browser extension that could take a Uniprot sequence and then find the correct gene sequence on NCBI, that would be great. Okay, what else we got? Somebody's asking for homework help. Is there a website for the thing that you're asking for? I mean, no. There's some of it, but it would depend on the gene, and it would depend on a whole bunch of other things. It's not really the right, like, it's nothing magic like that. You could, like, blast certain parts of it. You could use Benchlink for the reverse translate, but for identifying the exons is hard. Fave anime. That is a hard. That's a hard. It's a hard thing to pick. There's a lot I like. Gate is pretty good. Shokugeki no Soma is fantastic. I mean, then the usual Full Metal Alchemist was really good. Attack on Titan was middling. I don't know. Seth asks, I'm on your GitHub. How do I copy the milk and eggs plasmid into Benchlink? I assume I save it as a file and upload. Yeah, that's it. That's exactly what you do. In fact, here I can show you. So yeah, let's say milk and eggs, for example. Where is the fucking download? How do I download? Dude, I suck at GitHub. Edit this file. Open this file. GitHub. Yeah. Come on. How do you download? God, I suck at GitHub. Yeah, you download the file, which is apparently fucking difficult. How do you download shit? Copy link address, save link as. If I click that, what happens? GeneBank file. Yeah, alright. Let's see what that does. Let's see if this works. Milk and eggs. Import. Choose. I'm just seeing if this works. I don't know if it will, but we'll see. Come on. Fucking trackpad. Seems like it's working. It did not like it. An unexpected error occurred. Woof. Okay. Wait, did it work? Okay, wait, I'm gonna just make a new folder, and I'm gonna try and do it that way. Because, junk. It might have just not liked that there's an identical sequence, so we're gonna try that again. Open. Raw usually triggers a download. Nope, it did. Okay, shit. Okay, how do I... Okay, so I click this. You said click raw? Where do you see raw? I don't see raw. Oh, raw. There we go. That's not better. Ah, shit. String. Come on. Fucker. I don't know. I have no idea what I'm doing. Okay, there's a browser extension. file save. I clicked raw. The problem is raw also gives you all this crap, which is... Oh wait, I wonder if I could just make this as like a notepad file. And then just save it as a GeneVac. And that's basically what it is. Okay, so let's click raw. This is not the right way to do this. I'm just mostly fucking around. Notepad. Paste. I don't know if this is gonna work. We're going with it. Downloads. Milk.gb. All files. Save. I have no idea if this is gonna work. I'm just testing this out. I don't think this is the right way to do this. You should almost definitely download the frickin' browser extension that lets you just do this, but let's see if this works. Or if this gets really angry. Yeah, it does. That's the dumbest thing I've ever seen. Okay. Yeah, there we go. That's how you do that. You go to raw, make a notepad, like open notepad, paste it in, change it to a .gb file, and then it does it. It just knows how to read that, apparently. There we go. We solved the mystery. Yay. What was that? So yeah, to Seth, that's how you do that, apparently. Now we know. Now we've learned together. PC build hardware list. Okay, I need to open something to do that, so I'm gonna set this to camera only. Give me a second and I'll see if I can dig it up. Just gonna make sure that that's switched over on the thing. Do you frequent any BIOS subreddit or Discord communities? The only subreddit that I frequent that's like science, well, I mean, other than like r/biology, is r/labrats, or r/labrat, because it's hilarious. But yeah, other than that, no. So I'm just trying to pull up the build. Where the hell did I put it? So yeah, this will just take me a second. I'm just trying to find my okay, that'll do. It's not really what I want. What was the name of this site? I should be able to find system builder. Maybe it saved? It did save. Yay! Exciting. Okay. I can switch this back to scene now. Okay. Cool. So yeah, this is my list. This is the bits. I decided I'm gonna do caseless, so I'm gonna build like a panel with everything, and I might put some lights on it or whatever. And this isn't the final list. I decided I wasn't gonna do the solid state drive. I might just add it later. But yeah, this is my build list so far. So we've already done the processor. I already have the motherboard. Now we've just covered RAM. So we're missing the cooler, hard drive, the graphics card, and then the power supply, and the laptop, or not laptop, monitor rather. But yeah, so we're working our way through it. We've got, I think I'll probably go for graphics card next, because I think that is the last. This I also found cheaper. I found this like half price. So I think I'll probably do graphics card next because that's the last like big chunk. And then the rest of it I might just order. Yeah, so that'll be really good. This is gonna be a really dope computer once it's all done and built. Yeah, I may still buy the solid state drive to go with the the proper hard drive, just to have my operating system on the solid state and then all my storage elsewhere. Um, just because it's meant for processing video, so I need a huge amount of internal storage, um, for it to work properly, and so we'll see. But I mean, there's multiple hardware or hard drive slots. I might even build a little RAID array at some point. All options. Um, Suggestions to go for a 7200 RPM hard disk. Okay. Bennis, no, I will not stop streaming after I get my PC. That is not the point of streaming. That's just the most recent thing we're fundraising for. We've done fundraisers for getting some of these pieces of DNA made. Like, literally this piece that I just showed how to get in here. This is currently being synthesized, and it's been paid for by many wonderful people who chipped in, donated. So, like, yeah, I'm gonna keep streaming. So also this is actually a good moment to address something which I've gotten some comments about, where people are like, I don't like this streaming. Why don't you go back to making videos? This is how I picture them in my head. To which I say, I'm still making videos. I'm literally, like, the rest of my day is editing a video I've been working on for literally months. So I'm going to continue to stream because I like doing it. I like this interactive element. I like getting to know some of the many people who continue to show up and be very helpful. And I've gotten great practice doing genetic design, which I have no reason to make a frickin' AAV with SYN thing that modifies magnetoreception. There's no reason I would do this on a regular, normal day. I have other shit to do. But doing the stream has been an excuse to design crazy, weird, wonderful things. And, some of them we end up getting made. Like the milk plasmid and the egg plasmid. And the egg plasmid is going to be used for the silk project later. So, like, I'm still going to be doing both. I like streaming. I've really come to enjoy it. I love this interactive element. I like that it's no pressure, and I can just talk about whatever the fuck I feel like, and have some of my cool friends on and talk about interesting things. Like, I like that we got to do the THC video a couple of days ago. Like, I got to do an interview with a guy who, there's no reason that he would be doing interviews like that, and I could ask him very technical questions, let you guys ask him some questions. So, yeah, that's a thing. So I'm gonna keep streaming, but I'm also gonna continue making videos. My current video schedule is to try and get one video per channel per month. So one food video, one science video, and I've mostly got my science stuff got delayed because the fucking pandemic hit, and so it screwed with a bunch of my orders that I've been waiting on for months, but they finally arrived. So, like, I'm moving forward on science stuff. Food stuff I've already got planned out. I think I know what my next video is going to be. Originally it was going to be a recipe from Shokugeki, but now I think I'm going to do a Jacques Pepin recipe because I love doing it and it's delicious. And the Shokugeki recipe was actually kind of underwhelming. Like, it was good, but it was a lot of work and kind of eh. But yeah, so yeah. Okay, let's do one more question, then I'm going to wrap it up for the day because I'm tired, my voice hurts, and I have a script to finish writing and record today. Yeah, I mean, the fact that, like, the viewage has really petered off after the start, once I finished, doing my intro, that's fine. I mean, not everybody's interested in seeing how I actually, like, tinker with DNA and the kind of nitty-gritty of parsing it and going through it, and that's fine. That's not really why I do it. I mean, I leave the videos up so that if you want to learn how to do this stuff, then, the resources there. Like, I'm not expecting, like, that's the other thing. Like, somebody sent me an email, like, just so you know, and of course this is somebody whose YouTube channel has like 50 subscribers, and they're like, just so you know, you're nuking your ability to get views on your main videos because you're streaming. And I'm like, okay, first of all, who the fuck are you? Second of all, no, that's not how that works. And, like, third, I don't give a fuck. Like, I enjoy doing the streams. They're fun. Like, oh no, it affects my viewage on my other videos, which, by the way, it doesn't. But, who cares? This is fun. Okay, so last question, and then we're going to wrap it up. It says, what are you planting behind you? So I think I counted 38 different species growing at the moment. But I'm going to just go through them and we're going to see what we get. So we've got, so there's actually more plants on the other side of the camera. I'm just not going to turn the camera around because I can't. So I'm going to just work from one side to the other, and I'm only going to count ones that have actually sprouted because I've planted some that just haven't sprouted yet. So I've got pink okra, I've got eggplants, I've got 2 species of strawberry, I've got orchid cream nasturtiums, I've got some dill, I have some lantanas, hibiscus, marigolds, curry plant, not curry leaf, like the curry flower, which is not edible, but it smells nice. Then I've got lettuce, I've got what's called spinach strawberry, which is weird. I've got marjoram, stevia, shiso, anise, peppermint, lavender, parsley, coriander, mimosa pudica, tarragon, sage, lemon balm, chives, and 2 species of onion. And then I'll also do a tray of microgreens from time to time. But yeah, that's all the plants I've got growing at the moment. So yeah, like I said, that's where we're gonna end it for the day. I hope you guys have enjoyed. Thank you so much to everyone who's donated. This has been a lot of fun. We're gonna finish up the design of this particular plasmid on the next Who's Gene, so next Tuesday. If you've got suggestions for a future episode of Who's Gene, leave them for me on Twitter. Twitter handle below. Again, a reminder, there is a new video on the Taste Emporium. This is the Taste Emporium. Go subscribe to the Taste Emporium. Please go subscribe to the Taste Emporium. So yeah, that's all coming up. There'll be a new Taste Emporium video in a couple of weeks. There should be a new video on my main channel, so here, in a week or so. I'm literally just editing it. It's going to take me a few days to finish up the edit, and then that'll be good. Fingers crossed it's the last video I have to edit on this laptop, but we'll get there. Yeah, so thanks for stopping by. On Friday, like I said, there's going to be a new biohack chat with Gabriel. That's going to be a lot of fun. We're going to be talking about permaculture, so people who like plants, finger guns, show up. So yeah, just so if you want to send me ideas for future streams, use the hashtag Who's Gene and just, at me on Twitter. My Twitter handle is Emporium Thought because some bastard has Thought Emporium. And yeah, so that's that. I'll just set this to camera. So yeah, thanks again for hanging out. This has been a lot of fun. I love doing these streams. I look forward to doing many, many more. Thanks again to the people who donated, and I will see you on the next one. Bye-bye. Have a good day. Be safe. Don't lick doorknobs. Bye.